First, we have Dr. Jason, and he's actually currently in Portugal. He is a postdoctoral research fellow, and he focused on application of nanobiotechnology, especially in diabetes. And it's quite amazing that he has authored about 25 book chapters. He wrote about 33 journal articles, and also he edited a book for Nova Science Publisher. And today he's going to share with us on the emerging nanomedicines for diabetes treatment. So let's welcome him.
This presentation is divided into 4 parts. In section 1, I'm going to introduce about nanomedicine. In section 2, I'm going to introduce about diabetes and its conventional treatments and how nanomedicine is going to be helpful. In section 3, I'm going to mention about my research projects, and in section 4, I'm going to mention about my current projects and invitation to collaborate. Let me start the presentation. So, introduction to nanomedicine. What is nanomedicine? As we all know, particles with the size of 1 to 100 nanometer is considered as nanoparticles. In certain cases, 1 to 1000 nanometer sized particles are also considered as nanoparticles. Similar to nanoparticles, medicines in the size of 1 to 100 nanometer or 1 to 1000 nanometer in size is known as nanomedicine. So what is nanomedicine? If you take a conventional medicine and fabricate into nano-sized medicines, it is termed as nanomedicine. Or if you have a conventional medicine and if you formulate it into a nano-sized particle, it is also considered as nanoparticles. The basic criteria here is if you reduce the size, its property should differ. So that's the only criteria to call them as nanomedicine. For example, in the first case, the example is if you have nanomedicine, for example, doxil or metformin or something, and if you fabricate them into a nanoparticle, nano-sized metformin, nano-sized doxil, which is called as doxorubicin. So those things are called as nanomedicine. In other terms, if you have a metal or polymer or a metal oxide nanoparticle and formulate them into a nano-sized liposome or micelles, it is also termed as nanomedicine. So what is the significance of this nanomedicine? Most of the biological macromolecules such as mitochondria, ion channels, secretory granules, lysosomes are nanostructures. So it is easier for these nanomedicine to target these nanostructures, engineer them, repair them without having any side effects, and also have more bioreactivity. So that's why these nanomedicines are used as controlled and target drug delivery system in bioimaging, in biosensors, in biomarkers, biomarker mapping, gene delivering, molecular imaging, and in several biomedical and pharmaceutical applications. This slide shows the history of nanomedicine. The history of nanoparticles started in 1905 when Einstein estimated the size of sugar compound as 1 nanometer. And it took about 90 to 100 years for Sumio Ijima to invent carbon nanoparticles in 1991. And in 2000, many countries have developed their nanoforums to develop projects on nanoparticles and everything. And in 2009, nanoparticles have been used as a targeted drug delivery system to cancer tissues in vivo condition in mice models. And from 2016 until now, more studies are moving towards nanotoxicology as nanoparticles and nanomedicines are entering into commercial space, and more it can affect environment or human or other living organisms. So that's why currently it is moving towards this phase. This slide shows the history of nanomedicine in brief. From 1964 to 1994, this is considered as an era of conceptualization of nanomedicine. In this time period, liposomes, dendrimers, polymeric nanoparticles, micelles, everything was considered as the materials or agents to formulate nanomedicine, or conventional medicines formulate into these type of polymers. So that conceptualization era has led to the clinical validation and commercialization era from 1995 to 2007. So this area, in this time period, many particles have been developed and they have also clinically validated, and they move towards a commercialization and FDA approval and so on. From 2008 to 2011, this clinical validated nanoparticles or nanoformulations as nanomedicine has been expanded and refined for several biomedical and pharmaceutical applications. And from 2016, it has been moving towards nanotoxicology era. Why nanomedicines are special? Why researchers are highly motivated towards nanomedicine research? As I said before, to call a medicine as nanomedicine, it should be under 1 to 100 nanometer in size, or 1 to 1000 nanometer in size, whatever, but its property should differ from that of a conventional medicine. This property difference is because of two aspects. One is high surface to volume ratio, which is related to its size. When you reduce a bulk material into a nanomaterial, its surface properties differs. For example, if you have a bulk material like this, only 4 atoms are exposed to the surface. But in the same volume of a material, you will have several nanoparticles since you reduce the size, and because of its size, you will have not 4, you have 14 or 16 or several hundreds of atoms which is exposed to the surface. Because of this high surface to volume ratio phenomenon, its bioreactivity is higher, its bioavailability, its biodegradability is higher, because of which nanomedicines are special. The second one is related to its shape. In this slide, I have shown the different shapes of nano zinc oxide nanoparticles. It can be fabricated into ring, into hexagon, into fibers, and several other shapes. And depending upon shape, also nanomedicine's property will differ. So that's why when the property of a nanoparticle, when the size and shape of a nanoparticle differs, its property differs, and because of which nanomedicines are highly fascinating for researchers to research on. In the previous session, I have summarized the importance of nanomedicine. So in this session, I'm going to introduce about diabetes and what are the conventional treatments and how nanomedicine can be helpful in the treatment of diabetes. So what is diabetes mellitus or diabetes? Every day we are taking food. Food is required to give you energy to do any activity. So once you take the food, it goes to the intestine where it gets digested, and because of the digestion process, it is converted into glucose, and this glucose is taken to all the parts of the body, all the cells of the body through blood. So this is how the glucose goes to all the cells, and this glucose should be converted into energy form, into ATP, which I will say in the later slides. So this glucose will be converted into energy, which is required for the cells to activate, to work, to provide you energy. So this glucose in diabetes, this glucose level will increase in blood, which will increase the viscosity of the blood, and because of the increase in viscosity, this will affect the flow of blood, and which will cause a block in blood vessel or blood pressure. So in general, diabetes is a disease where blood sugar level, blood glucose level increases for a prolonged time period, not for a short time period, for a prolonged time period. So it should be for at least 6 months or 1 year, it should be in a rise, it should increase. So why do we need to worry about increase in blood glucose level? because increase in blood glucose level will lead to block in blood vessel or increased blood pressure. So in Asia, in average condition in Asia, before meal it should be the blood glucose level should be 4 to 5.9 millimole per liter, and after meal it should be below 7.8 millimole per liter. And if this level is increased for a prolonged time period, it is called as diabetes mellitus. In general, diabetes is classified into two types. One is type 1 diabetes and another one is type 2 diabetes. In type 1 diabetes, the body's inability to produce insulin. So your insulin, the pancreas will produce insulin. So your pancreas will have a malfunction. So because of which insulin will not be produced in these type of diabetes. But in type 2 diabetes, insulin will be produced, but your cells cannot respond or fail to respond to insulin properly, which is a condition called insulin resistance. So that is type 2 diabetes. In type 2 diabetes patients, insulin will be secreted, but it cannot be utilized by the cells. And there is a third type, which is gestational diabetes, which is also similar to type 2 diabetes, but it occurs only in pregnant women during pregnancy. So what is insulin, its function, and insulin resistance? We will see in next slide. Insulin is a peptide hormone, a protein in the endocrine system produced by beta cells of pancreas. This figure shows the mechanism of insulin. As I said before, after you intake your food, it goes to the digestive system. In intestine, it gets digested and it gets converted into glucose, and it is taken to all the parts of the body or the cells of the body through blood. So once the glucose enters into the blood, insulin will be secreted by the pancreas, and it is also taken to all the cells of the body through bloodstream. So from the bloodstream, insulin will also enter into your cell. So this glucose meets the insulin and gets converted into pyruvate. Pyruvate will be converted into fatty acid, and fatty acid to ATP, adenosine triphosphate. So this ATP gets stored in the mitochondria of the cells, which is powerhouse of the cell. And from this mitochondria, a cell will get energy for its function. So for the conversion of this glucose, insulin should be present inside your cell. As I said before, in type 2 diabetes, this insulin will be secreted, but it will not enter into the cell as there will be a block in this insulin receptor. So this term is called as insulin resistance in type 2 diabetes. This slide shows the timeline of anti-diabetic drugs developed throughout history. In 1923, insulin has been developed, and from 1923 to 1956, several insulin types has been developed to improve their efficacy. And later in 1956 and 1959, metformin and tolbutamide has been produced, and this type of drugs has been used to degrade glucose into ATP in the presence of insulin. So it goes along with insulin. So it's like a combination treatment for diabetes. But this will lead to hypoglucose condition, so reduction in glucose level in the blood, so which will lead to anemic condition and so on. Later in 1955 to 2013, several drugs has been produced, which is to convert this glucose into ATP without the presence of insulin for both type 1 and type 2 diabetes patients. But all these drugs has led to one type of side effect or another, like increase in obesity or increase in blood pressure or decrease in blood pressure or increase insulin condition or insulin or damage in the cells and so on. Because of the drawbacks in conventional drugs, several researchers propose that nanoparticles or nanomedicines can be beneficial for the treatment of diabetes. Swinnen et al. In 2009 showed that insulin therapy or treatment with conventional drugs to convert glucose into ATP can lead to hypoglycemia, which is decreased blood glucose level, and weight gain. This is because in all these studies they have used high concentration of insulin or conventional drug in those treatment models. So that's why nano formulations have been used for the controlled delivery of insulin. For example, pluronic F127 for buccal insulin delivery, insulin-loaded polymeric nanoparticles for oral insulin delivery, chitosan nanoparticles for oral insulin delivery, and chitosan-reduced insulin-loaded gold nanoparticle for nasal delivery of insulin has been used for the controlled delivery of insulin. Until now, all the nanoformulations have been used only for controlled delivery of insulin. This type of treatment can be useful only for type 1 diabetes. As I said before, in type 2 diabetes, the problem is insulin resistance and not insulin secretion. That's why in my projects, I'm using a nanomedicine as a supplement to reverse this insulin resistance. So I will explain about my project in the next session. In session 3, I'm going to explain about nanomaterials to reverse insulin resistance in type 2 diabetes treatment. Nanoformulations have been useful for controlled delivery of insulin, as I mentioned in my previous sessions. But nanoparticles as nanomedicine has been useful for certain diabetes treatment applications. For example, gold nanoparticles are useful for bioimaging of diabetic cells, and gold nanoparticles are also useful as a glucose sensor, as a biosensor for monitoring of glucose in diabetic patients. Silver nanoparticles, for example, they are useful for antimicrobial activity, especially in diabetic foot ulcer treatment. But the limitation of these metal-based nanoparticles, they are not stable and they get oxidized easily. For example, silver can be formed into silver oxide. So that's why we prefer metal oxide nanoparticles compared to metals, as they are stable and they are biologically active and compatible then and biocompatible than metals. However, most metal oxide nanoparticles are used only for insulin delivery, as I mentioned before. For example, copper, zinc, manganese, titanium, and cerium oxides. And composites and biometallic nanostructures are used in glucose biosensors. Why magnesium oxide? So in my work, I'm going to use magnesium oxide nanoparticle as a magnesium supplement to reverse insulin resistance. So why magnesium oxide? Kandil et al. In 1996 showed that magnesium sulfate can reverse insulin resistance. But in that work, he used high concentration and it takes 48 hours. And May et al. In 2004 showed that high concentration of magnesium can cause cell proliferation. It can lead to cell development. And Probagallo et al. In 2003 showed that insulin resistance due to low magnesium level. There is a correlation between insulin resistance and magnesium level. And he also mentioned that magnesium is needed for the activation of enzymes, but he didn't mention which enzyme. And G et al. And several other studies show that magnesium oxide nanoparticles are non-toxic to human cells and magnesium nanoparticles are stable in body fluids. Because of these reasons, magnesium oxide nanoparticle has been selected to reverse insulin resistance in type 2 diabetes. So the aim of my project was to study insulin resistance reversal ability of non-toxic MgO nanoparticles as magnesium supplement to treat type 2 diabetes. In the previous study, Kandil et al. In 1996, so consider this one as an insulin resistant cell, diabetic cell, right? So in that work, he used magnesium sulfate, and this magnesium sulfate will bind with the surface of the cell and it disintegrate into magnesium plus and sulfate ions, which will activate extracellular enzymes. But in the current study, in my project, I've used magnesium oxide nanoparticle. So this nanoparticle can bind with the surface of the insulin resistant cells and also enter into the cells, into the diabetic cells, and activate both extracellular and intracellular enzymes and reverse insulin resistance and make insulin to enter into the cell. So glucose will also enter into the cell, so it will convert glucose into pyruvate, pyruvate to fatty acid, and fatty acid to ATP, so the cell will get energy. So a diabetic cell will be converted into a normal cell in type 2 diabetic patients, provided they have perfect insulin secretion. So the significance of the study is the synthesis methods in my study has yielded different morphologies. I have used chemical synthesis method as well as green synthesis. In chemical synthesis, I have used Sol-Gel method, and in green synthesis, I have used aqueous extract of certain plants. And morphologies like hexagon and rod shape can facilitate entry of nanoparticle into cell. So different morphologies included spherical, hexagon, and rod, and we have identified that hexagon and rod shape can facilitate entry of nanoparticle into cell. So in this hexagon, hexagon is transformed into an extended hexagon, which is in a type of rod. So I will mention about later, or you can read our previous manuscripts. Cytotoxic study will help in screening toxic nanoparticles. So we have used double stage cytotoxic analysis by using diabetic cell lines, 3T3L1 cells, as well as Vero cell lines, kidney cell lines. So normal cell and diabetic cells. So in both these cell lines, the selected nanoparticles are non-toxic or less toxic. And the major significance is the development of nanoparticle that can reverse insulin resistance without any side effect, as we are going to use only a few doses of nanoparticle and it takes only 24 hours. As magnesium oxide is synthesized in nanoform, less concentration will be needed for reversing insulin resistance. So the novelty of the study, development of different morphologies is first novelty. So we have synthesized spherical, hexagon, and rod by using chemical synthesis, and we have developed spherical and hexagon shaped nanoparticle by using green synthesis methods. Previous studies focused on spherical shaped MJ nanoparticles using Sol-Gel, but in our method we have produced hexagonal and rod shaped MGO, which has the ability to penetrate into cell and activate enzymes. And green synthesis of magnesium approach to reduce toxicity. So this is the first time we have synthesized magnesium oxide nanoparticle by using a green aqueous aqueous crude extract of certain plants, Andrographis paniculator, green spinach, and red spinach. And this is the first work to use nanomaterial to reverse insulin resistance. So this is the major novelty of the present study. So here is the results of our study. You can see our results in these two articles. So this shows the cytotoxic analysis MTT assay in both 3T3L1 and Vero cells. Sample B is Sol-Gel synthesized hexagonal shaped MGO nanoparticle, and this is sample GA is from Andrographis paniculator. From Andrographis paniculator, sorry, not Andrographis paniculator, like Amaranthus tricolor, the red spinach one, and it has been transformed from spherical to hexagonal by using pH alterations. Right, so the hexagonal shaped green synthesized MGO nanoparticles, the results are present here. So this shows that in diabetic cell lines they are less toxic, but in normal cell lines they are almost non-toxic, and especially green synthesized ones are less toxic compared to the Sol-Gel synthesis one. The main important result is here. So compared to the control, the green synthesized MGO nanoparticle has reduced both the green synthesized and the chemical synthesized nanoparticle both reduce the glucose present in added in the diabetic cell lines with the help of insulin. And this takes only about 25 hours to reverse insulin resistance. To reduce the glucose by reversing insulin resistance, and and the Western blot analysis also showed that GLUT4 protein has been expressed uh, in those uh, in those samples. So you can read the results in these two papers. These are the news articles where our research has been acknowledged. This is a news article in Malaysia. Innovative Malaysia in the Star newspaper, they have acknowledged our research, and in several platforms as well as internet news articles. These are the research articles which we have published related to our project. Um, and we are also currently working on aptamer navigated copolymeric drug carrier system to deliver these MgO nanoparticles. So our idea is to encapsulate these MgO nanoparticles to target only diabetic cells, to reach the diabetic cells, release MgO nanoparticles, and reverse insulin resistance in only in diabetic cells and leave out normal cells. So our current projects and possibility for collaborations. So currently I'm working on dendrimer formulated MgO nanoparticles for biological applications. So since MgO nanoparticles have been working for about 5 years, so currently I'm going to work on to stabilize these MgO nanoparticles by using dendrimer formulations, and also to use them as a drug carrier system to reach diabetic cells or cancer cells, to reverse insulin resistance, or as an anti-cancer ability. So currently I'm working in University of Madeira. It is in the city of Funchal, Madeira, Portugal. It is a Portugal government university. For those who doesn't know, Portugal is here and Madeira is in the center of Europe and Africa. So we are here. It is a small beautiful island. And this is our main campus, which is built in 16th century, and this is a new campus which has been built in 1988, and our department is in the ground floor of this building. And this is our center, CQM, Centro de Química de Madeira, and I belong to Molecular Materials Research Group. The core research area of this group is dendrimer formulation for drugs and nanomedicine, and we also synthesize carbon nanomaterials. And currently I'm working on dendrimer formulation of MgO nanoparticles for both cancer, for anti-cancer activities, as well as diabetic treatment. And we invite all young collaborators, so those who are working in diabetic cell lines or animal models, so those who want to work in this field to test this, to help us to test our nanoparticle or dendrimer formulated nanoparticles, you're always welcome to contact me. My other research interests are we are also developing and evaluating glucose responsive nanogels for type 2 diabetes, and this is a collaborative project with Biolim Limited Bangalore. It's a company, it's a small scale biotech company in India, and we are also working on nanoparticles as light mediated antibacterial paints. So there are two projects in chemical engineering department, Curtin University, Malaysia, my alma mater, and antibacterial study of charge and concentration specific purified endotoxin towards E. coli and Pupitida. This is a collaborative project in University Malaysia Sabah, Malaysia, and we're also working in novel coronavirus biosensor using aptamer. It is a in silico model. We are working on in silico models with Professor Michael in University of Tennessee at Chattanooga, USA, and we are also working on drug repurposing for treatment of COVID-19, which is a project funded by Indian government with ACRT Biotech. It is also a small-scale biotech company in India. So about me, I am Dr. Jason, as you all know. I studied, I did my bachelor's in zoology and biotechnology, and later I moved towards master's in nanoscience and MBA, master's in business administration. Human resource management is my area. All these until master's, I did my studies in India, and then I moved to Malaysia, where I did my PhD in Curtin University, Malaysia. Malaysia, and the project which I was working on and still I'm working in was funded by Curtin University, Australia. And yeah, so all the results and everything was from that time period. And I have experiences in nanoparticle synthesis, especially green synthesis of plant particles, of plant extracts, characterization of nanoparticles, cytotoxicity analysis, and as well as in vitro diabetic studies. And current research, I am focusing on using application of bio nanotechnology for the fabrication and nanoformulation of drugs for the treatment of several diseases, especially diabetes and cancer. And till date, I have authored about 36 book chapters, 45 journal articles, and edited a book for Nova Science Publishers, and several conference papers. And my previous past careers, past research experiences are in Curtin University, Malaysia as a research associate, and MKD Labs with Professor Michael as a research consultant. And currently I'm also editing and writing certain books for Elsevier, United Kingdom, and currently I'm working as a senior researcher in University of Madeira. And these are our books. This book is currently available online. You can purchase them, and this book will be available in December 2021 or in early or probably early in 2022. So I would like to thank Biointegration Journal for the opportunity, Professor Saw, other conveners and coordinators of this Youth Forum 2021, and all the participants for your patience to hear this presentation. And this is my funding agency, so I'd like to thank them for funding my research. And this is my contact, email ID, blog, and my Facebook, how to reach me. And so thank you very much for your patience, and thank you. I'm looking forward for your collaborations.